Assalamualaikum and good afternoon. My name is Nur Adriana Natasha binti Abdullah, and today I would like to present my final year project proposal which focus on isolation and characterization of drought-tolerant bacteria capable of producing C04 from soil in Jelebu district. I will begin my presentation with briefly explain about the introduction. First is background of the study. Drought is defined as prolonged dry condition that significantly affect soil fertility and plant growth. Drought reduce soil moisture, nutrient availability and disrupt plant physiological process such as photosynthesis. In Malaysia, Jelebu district is known as the driest regions which often face drought stress due to the low annual rainfall. This suggests the presence of the drought-tolerant bacteria in the Jelebu district soil. Drought-tolerant bacteria that are capable of producing C04 act as a plant growth-promoting bacteria in helping plants to survive under drought stress. For the problem statement, Malaysia frequently experiences droughts that negatively affect agricultural productivity and lead to economic loss. Currently, chemical fertilizers are widely used to improve crop yield, but they are costly and environmentally unsustainable. Drought-tolerant bacteria producing C04 has a potential to be used as natural biofertilizer in the future as it can promote the plant growth. However, there are limited information regarding the presence of those bacteria in Malaysia, especially in Jelebu soil, that resulting in the opportunities to develop natural agricultural applications to improve agricultural productivity during drought in Malaysia are missed. Next is the objective of this study, which include to investigate bacteria from soil in Jelebu that producing C04, to evaluate the tolerance of bacteria to the drought conditions, and to characterize the bacteria based on their morphological and biochemical properties. The study is significant to conduct as it addressed the lack of information regarding the presence of those bacteria in Jelebu soil and provide new scientific information regarding those bacteria. This study provides better understanding of regarding bacterial characteristics which will contribute to the development of sustainable and natural agricultural applications such as biofertilizer in the future. For the research outcomes, it is expected to successfully determine the presence of drought-tolerant bacteria capable of producing C04 from soils in Jelebu district and identify their characteristics to acquire their potential for future use in agriculture applications. Next is the literature review that explains the scientific concept that related to my research. Drought is a prolonged dry condition that causes water scarcity, which reduces the soil moisture and nutrient availability, and also disrupt the plant process, leading to the reduction in a crop yield. Next, the drought-tolerant bacteria are beneficial soil microorganisms that can survive under low water conditions and stabilize the soil structure and also promote the plant growth. It consists of thick peptidoglycan layer and spores which help them to resist in drought stress. The mechanisms of these bacteria include the production of osmoprotectants, exopolysaccharides, biofilms, phytohormones, and antioxidant enzymes. Next, C04s are iron chelating compounds produced by bacteria that bind to iron and convert it into the soluble form. Under drought conditions, C04 can help in enhance the iron uptake to the plant, promoting the plant growth. For the research gap, despite numerous studies regarding these bacteria worldwide, limited research has been conducted in Malaysia and no specific study focused on Jelebu soil. Lack of data limits the development of future agriculture applications. Next is the methodology. This chapter highlights the method used to achieve the study objectives. First is soil sampling. Soil sample will be collected from 3 different locations at a depth of 5 to 10 centimeters. Then the sample will be packed into sterile plastic bags and stored at 4 degrees and will be processed within 24 hours. Second is preparation of soil suspension and cereal dilutions. Each sample will be weighed and transferred into the conical flask containing sterile saline solution. Then the mixture will be agitated for 30 minutes at 150 rpm. For cereal dilutions, 1 ml of soil suspension supernatant will be transferred into clean tube containing 9 ml of sterile saline. Then the solution will be diluted up to the 10 power of -6. Next is isolation of bacteria. 0.1 ml of diluted sample from dilutions of 10 power of -5 and 10 power of -6 each will be pipetted onto the nutrient agar and will be sprayed using a sterile glass spreader to ensure the uniform distribution of the sample throughout the entire agar. Then the agar plate will be incubated at 30 degrees Celsius for 24 to 48 hours. After incubation, the bacteria colonies will be observed. Next is purification of bacteria. The distinct colonies will be selected to subculture on the nutrient agar by the streaking method. Then the bacteria culture will be incubated at 30 degrees Celsius for 24 to 48 hours. Then the colonies will be observed for their identical characteristic to ensure pure bacteria isolates are obtained. Next is screening for siderophore production. The bacteria isolate will be inoculated onto the chrome azuro S agar and then incubate at 30 degrees Celsius for 48 hours. After incubation, the presence of orange halo around the colony will be observed, which indicate the production of siderophore. Next is screening for drought tolerance. Polyethylene glycol 6000 will be amended in 10 ml of nutrient broth with different concentrations, which is 5%, 10%, 15% and 20%. Then the bacteria isolate which produce siderophore will be inoculated into the nutrient broth with different concentrations of PEG 6000. Then the nutrient broth will be incubated at 28 degrees Celsius for 24 hours under constant shaking conditions. After the incubation, the bacteria growth will be measured using spectrophotometer to determine the degree of the turbidity of the solution. Next is morphological characterization method, which is gram staining. Bacteria colony will be smeared onto the clean glass slide and the slide will be dried on the hot plate to fix the smear. The slide will be stained first with crystal violet for 60 seconds and then stained with iodine solution for 60 seconds, decolorizer for 5 seconds and counter stain with safrani for 60 seconds. For each staining step, the slide will be rinsed with water first before proceed to the next stain. After done staining and dry the slide will be examined under the microscope to observe the bacteria cell morphology such as shape, color and arrangement. Next is biochemical characterization method. First is catalyst test. Hydrogen peroxide will be added onto the clean glass slide and bacteria colony will be added. Bubbles formation will be observed immediately and the formation of the bubble indicate positive result while no bubbles indicate negative result. The second test is indole test. Bacteria colony will be inoculated into the peptone water and incubate at 30 degrees Celsius for 24 hours. After the incubation, Kovacs reagent will be added into the culture and the formation of red ring will be observed. The formation of red ring indicate the positive result. Third is the citrate test. Bacteria colony will be streaked onto the Simon citrate agar land surface and incubate at 30 degrees Celsius for 24 hours. The color changes will be observed. Blue formation indicate the positive results. Next is data analysis. All experiment data will be recorded systematically and for the group of bacteria under different concentrations of PEG 6000, it will be analyzed by using one-way analysis of variance. This Gantt chart outlines the timeline of my final year project. The schedule ensures that all experimental work and analysis can be completed systematically. And this is the list of references that support the methodology and scientific basis of this study. That's all from me regarding my final year project proposal. Thank you for your attention.